Welcome to Last Minute Lecture. This free chapter overview is designed to help students review and understand key concepts. These summaries supplement, not replace, the original textbook and may not be redistributed or resold. For complete coverage, always consult the official text.
Right now, your brain is actively deleting, probably 99% of everything happening to your physical body.
Oh, easily.
Like the feeling of the shirt resting on your shoulders or the hum of the refrigerator in the background.
The pressure of the chair you're sitting in.
Exactly. It's all just being trashed by this incredibly aggressive biological spam filter. But if someone were to, say, drop hot coffee on your arm...
Right, that filter completely overrides the system.
Yeah, in a fraction of a millisecond, it routes the signal directly to your motor regions and forces you to yank your arm away before you even consciously realize you've been burned.
It is a ruthless prioritization system.
Yeah.
If you didn't have it, the sheer volume of sensory data would it would instantly overwhelm you.
You'd be paralyzed by noise.
Completely paralyzed.
Well, if you are a student staring down your first medical physiology exam, we've got you.
Welcome to this deep dive brought to you by the Last Minute Lecture team.
Glad to be here. Today, we have a very specific, very vital mission. We are taking on Chapter 46 of the Guyton and Hall textbook of medical physiology. So we're talking about the organization of the nervous system, the basic functions of synapses and neurotransmitters.
Which really is the bedrock of neurophysiology. Understanding the logic of this specific material is, well, it's the only way the rest of the nervous system will make any sense to you.
Right. And here is our promise to you. We are following the strict, unbreakable logic of the source material. We are translating dense mechanisms into plain English, and we are not bringing in a single outside distraction.
No tangents today.
No tangents. We're building a chain from basic microscopic anatomy all the way up to integrated physiological outcomes. So let's start at the macro level design. We are dealing with, 80 to 100 billion neurons in the central nervous system?
Yeah, it's a staggering number.
How do we even begin to organize that map?
Well, we split the entire architecture into two massive highways. First, you have the input pathway, the somatosensory axis. Imagine your sensory receptors. So things for pain, touch, temperature acting like millions of scouts deployed at the far edges of your body.
Right.
They capture information and send those signals up through the spinal cord, into the brainstem, and eventually all the way up to the cerebral cortex.
And then you have the output highway, right? The skeletal motor nerve axis.
Exactly.
And this flows in the exact opposite direction. So commands originate in the higher brain areas, travel down the spinal cord, and exit out to the effectors.
Right, and effectors are just the anatomical structures that do the actual physical work, like your skeletal muscles or glands.
So input and output.
Yeah.
But what happens in the middle is that integrative function we just talked about, that aggressive spam filter deciding what actually matters.
Yeah, and that routing happens across three distinct physical levels of the central nervous system.
Right. So let's walk through those three levels. First, you've got the spinal cord. And, I think the temptation is to view the spinal cord as just a dumb cable.
Oh, for sure.
Just a bundle of wires connecting your hand to your brain.
A lot of people assume that. But the spinal cord actually has its own highly organized local processing. Like, if you were to completely sever the spinal cord from the brain.
Which is a terrifying thought.
It is, but the circuits inside the cord are still fully capable of executing complex movements. They run your walking reflexes, your withdrawal reflexes from pain, and even local control of your blood vessels.
Wow. So it's handling the immediate physical reactions entirely on its own.
Yes.
Okay, so the second level is the lower brain, or the subcortical level. We're talking about structures like the medulla, pons, hypothalamus.
Right.
And this level manages the subconscious activities that just keep you alive.
Yeah, your arterial blood pressure, your breathing, equilibrium. It even controls basic feeding reflexes, like salivating, as well as core emotional patterns like anger or pleasure.
Which leaves the third level, right? The higher brain, the cerebral cortex.
The big one.
The source material describes the cortex as this massive memory storehouse. It's where the nuance lives. Because without the cerebral cortex, the lower brain functions are, they're sloppy and imprecise.
Yeah, the cortex adds that fine-tuned precision.
But the relationship actually goes both ways, right?
Right.
The massive memory bank of the cortex is kind of useless on its own.
It is. It requires the lower brain to constantly wake it up.
Right.
The lower brain initiates the state of wakefulness, which basically unlocks the cortex so you can form conscious thoughts.
It's so interesting when you look at the block diagrams for this whole architecture. The textbook maps the nervous system almost exactly like a computer.
It's an eerie parallel, honestly.
It really is. You have input circuits functioning as your keyboard and mouse, output circuits acting as your screen and speakers. You have a hard drive, which is your memory.
Yeah, and then the brain acts as the central processing unit, the CPU, just constantly directing your attention from one thought to another, prioritizing whatever the active tasks are.
Right, but if we want to understand how this supercomputer actually computes, we have to scale all the way down to the individual wiring. We need to look at the junction where one nerve connects to another.
The synapse.
Exactly, the synapse. So the source material outlines two major types of synapses, electrical and chemical.
Right.
Electrical synapses are essentially direct physical tunnels called gap junctions between two cells. The cytoplasm connects, and ions just wash freely back and forth.
Which makes them incredibly fast.
Super fast. They are perfect for synchronous firing, like making sure a group of cardiac muscle cells contract at the exact same millisecond.
Exactly.
But I want to push back on the design here for a second. If electrical synapses are instantaneous, why wouldn't the human body just use them for everything? I mean, the textbook is very clear that chemical synapses are by far the most common type in the central nervous system.
Well, it comes down to traffic control. An electrical synapse is basically an open door. Signals can wash back and forth in both directions, which is fast, sure, but it risks chaotic electrical noise just spreading everywhere.
Oh, like a feedback loop.
Exactly. Chemical synapses solve this by forcing one-way conduction. The first neuron, the presynaptic neuron, secretes a physical chemical. The next neuron, the postsynaptic neuron, receives it.
So the signal can only ever travel forward.
Yes. Chemical synapses act like biological traffic lights.
Okay.
They allow the nervous system to precisely route or delay or completely block information.
Okay, let's picture the anatomy of one of these connections, specifically looking at a typical anterior motor neuron in the spinal cord.
Yep.
It's built with three main parts. You have the soma, which is the main body of the cell. You have a single axon leaving the soma, which acts as the singular output cable.
Right.
And then you have this massive sprawling tree of branches called dendrites. And the scale here is just staggering. The text says there can be up to 200,000 minute synaptic knobs, presynaptic terminals, trying to connect to a single neuron.
What's even wilder is the real estate they take up. Like 80 to 95% of those 200,000 terminals are strictly connecting onto the dendrites.
Wow.
Only a tiny fraction connect directly to the soma itself.
So you have thousands of separate inputs all trying to whisper into the branches of this one cell.
Yeah, that's a good way to put it.
But for that whisper to happen, the terminal has to release its chemical payload. How does it know exactly when to do that?
For that, we have to zoom into the molecular choreography of what's called the active zone.
Okay.
Inside the presynaptic terminal, you have hundreds of tiny bubbles called vesicles. These vesicles are just packed full of neurotransmitters.
And they are just waiting at the edge of the membrane.
Exactly, waiting for a voltage change. So when an electrical signal, an action potential, travels down to the terminal, it depolarizes the membrane.
Right.
That sudden shift in electricity causes specific voltage-gated calcium channels to just snap open. And calcium ions from outside the cell absolutely flood into the terminal.
And the volume of neurotransmitter released is directly mathematically tied to the number of calcium ions that enter, right?
Yes, exactly.
Calcium is the trigger mechanism.
It is. The calcium binds to a special receptor protein on the vesicle itself called synaptotagmin.
Synaptotagmin.
Right, which kicks off a mechanical interaction with a group of proteins called the SNARE complex.
Okay, I always picture the SNARE complex like docking a supply spacecraft at the International Space Station.
Oh, that's perfect.
You have this vesicle floating there. It has mechanical arms reaching out of V-snare called synaptobrevin, and the cell membrane has receiving arms reaching back T-snares called syntaxin and SNAP25.
And those arms have to interlock perfectly.
Exactly.
And when that calcium binds to the synaptotagmin, it acts like the final command code. It violently pulls those SNARE proteins together, forcing the vesicle to fuse with the cell membrane and dump its chemical contents into the gap between the cells.
Excitatosis. The payload is delivered into the synaptic cleft. Now, the receiving cell has to catch it. The postsynaptic membrane is studded with receptor proteins, which generally fall into two categories: ionotropic receptors and metabotropic receptors.
Right. So ionotropic receptors are built for speed. They are direct ion channels. The neurotransmitter binds to the outside, the gate physically swings open, and ions rush in within a fraction of a millisecond.
It's just boom, open.
If it opens a sodium channel, positive charge enters, exciting the cell. If it opens a chloride channel, negative charge enters, inhibiting the cell.
So that's an instantaneous reaction. But sometimes the body needs a prolonged effect. Like if you're forming a memory, you need a change that lasts seconds, minutes, or even years.
Yeah, you can't just leave an ion gate propped open for a month.
Right. That's where the metabotropic receptors come in. These don't have direct ion channels at all. Instead, they activate an internal second messenger system.
And the most prominent example the source text provides here is the G protein mechanism. Okay, walk us through that. Picture a complex sitting inside the cell, attached to the bottom of the receptor. It has 3 parts: an alpha, a beta, and a gamma subunit. Normally, it's inactive, holding tightly onto a molecule called GDP.
But when a neurotransmitter binds to the outside of the receptor, the entire structure shifts.
Right.
The alpha subunit drops the GDP, grabs a highly energized molecule called GTP, and literally severs itself from the beta and gamma parts.
Exactly. The alpha subunit is now active and completely free to wander through the cytoplasm, where it can execute one of 4 prolonged tasks.
Okay, what are the tasks?
First, it can go find a specific ion channel from the inside and physically hold it open for a long duration.
Got it.
Second, it can activate internal chemical amplifiers like CANMP or cGMP. Third, it can trigger intracellular enzymes that completely change the cell's internal metabolism.
And fourth, which is the one that really blows my mind, the alpha subunit can activate gene transcription.
Yeah, it's incredible.
It literally travels into the nucleus of the neuron, interacts with the DNA, and commands it to start building brand new proteins. It physically changes the structural architecture of the neuron.
Which is wild, because that physical reconstruction is the foundational basis of long-term memory.
It's amazing. But this raises the question, what specific chemicals are inside those vesicles triggering these vastly different reactions?
Well, the source material groups the messengers into 2 vastly different shipping methods: small molecule transmitters and neuropeptides.
Okay.
Small molecules are for rapid acute responses, like dodging a moving car or blinking.
Right.
They are manufactured quickly right there in the presynaptic terminal cytosol. They get packed into vesicles, released, and then the vesicle membrane is actually reabsorbed and recycled back into the terminal to be used over and over again.
And the text highlights several heavy hitters here. You've got acetylcholine, which is crucial for the motor cortex and firing skeletal muscles.
Right.
Norepinephrine controls wakefulness and mood. Dopamine is generally an inhibitory signal. Glycine and GABA are your primary inhibitory transmitters in the spinal cord and brain. And glutamate is the major excitatory transmitter across the central nervous system.
Yeah, and then you have the weird rule breaker.
Nitric oxide.
Yes. Nitric oxide. It isn't stored in vesicles at all.
It's a gas, right?
It's a gas. It's synthesized instantly on demand, and it diffuses directly out of the terminal and into surrounding cells, altering their metabolic functions without even changing their membrane potentials much.
It's just a totally different mechanism. So contrast those fast, locally recycled small molecules with the neuropeptides.
Right. Neuropeptides are the heavy lifters for slow, immensely powerful actions. They aren't made in the terminal at all.
Where are they made?
They are manufactured all the way back in the main body of the cell, the soma, by the ribosomes.
Oh, wow.
Yeah. They get packaged by the Golgi apparatus, and then they have to be physically transported all the way down the entire length of the axon.
Axonal streaming. And the textbook notes this transport is agonizingly slow, like just a few centimeters a day.
It is so slow.
And when they finally reach the terminal, they are released in tiny quantities, and the vesicle isn't even recycled, it's just destroyed. So why would the nervous system rely on such a laborious shipping method?
It's all about potency and duration. Neuropeptides are often 1,000 or more times as potent as small molecule transmitters.
1,000 times.
Easily. And their effects, like closing calcium channels or fundamentally altering the metabolic machinery, can last for days, months, or even years.
So you use small molecules for split-second decisions, but you use neuropeptides to fundamentally adjust the system's long-term baseline.
Exactly. And the cool thing is, a single neuron doesn't actually have to choose between the 2.
Right, co-transmission.
Yes. Through co-transmission, a neuron can actually release both a fast small molecule and a slow neuropeptide at the exact same time. It sends an immediate command and a long-term adjustment simultaneously.
Okay, so we have the anatomy and we have the chemistry. Now we must translate that chemical release back into pure electricity. Because voltage is the only language the neuron ultimately uses to fire.
Right.
Now, I need to pause us here because this is where my eyes usually glaze over when reading physiology.
It gets dense, yeah.
The text states the resting membrane potential of a neuronal soma is exactly negative 65 millivolts. And I can memorize that number for a test, but what does it actually mean to have negative voltage inside a physical cell? Why specifically negative 65?
Well, to understand the math, you have to look at the Nernst potentials of the individual ions. A Nernst potential is essentially the exact electrical voltage that would perfectly satisfy a specific ion's desire to diffuse across the membrane, based on its concentration inside versus outside the cell.
Okay, let's use the leaky boat analogy.
I like this one.
Imagine the neuronal membrane as a boat floating in the water. The Nernst potentials are the specific water pressures constantly trying to leak in or out of the boat.
Right.
And the cell has active pumps acting like sailors frantically bailing water to keep the boat floating at a perfect depth of negative 65 millivolts.
That's exactly it. So the primary leaks involve sodium, potassium, and chloride.
Okay.
Sodium is highly concentrated outside the cell. It desperately wants to rush in. If sodium had its way, it would flood the boat until the internal voltage reached positive 61 millivolts.
Wow, okay.
Potassium is highly concentrated inside the cell. It wants to leak out until the inside hits negative 86 millivolts.
Right.
And chloride is highly concentrated outside, and its Nernst potential sits at negative 70 millivolts.
So sodium is pulling toward plus 61, potassium is pulling toward negative 86, chloride is pulling toward negative 70. How on earth does the boat maintain a steady negative 65?
Active bailing. The sodium-potassium pump constantly churns. It physically throws 3 sodium ions out of the cell while pulling 2 potassium ions back in.
It's fighting the leaks.
It fights those leaks continuously to hold that membrane potential perfectly steady at negative 65 millivolts.
But then a signal arrives. An excitatory transmitter binds to a receptor and forces the sodium channels open.
Right.
Suddenly, positive sodium ions rush into the cell. This neutralizes the internal negativity, pushing the internal voltage up from negative 65 toward negative 45 millivolts.
And that upward jump and increase of plus 20 millivolts is your excitatory postsynaptic potential, or EPSP.
EPSP, right.
If the internal voltage hits that negative 45 millivolt threshold, it triggers an explosive action potential.
But the action potential doesn't actually ignite in the soma where those synapses are located, right?
No, it doesn't.
It starts further down at the axon initial segment, the very base of the output cable. And why there? Because the soma barely has any voltage-gated sodium channels. But the axon initial segment is just packed with them. It has 7 times the concentration.
It is hypersensitive.
So the voltage has to physically spread from the soma to that initial segment to trigger the blast.
Which brings us to the crucial counterbalance. Because physiology isn't just about matching the gas pedal.
Right. If we couldn't hit the brakes, every minor sensory input would cascade into a massive full-body seizure.
Exactly. Enter the inhibitory postsynaptic potential, or IPSP. If an inhibitory synapse fires, it doesn't open sodium doors. It opens chloride or potassium doors.
Okay.
Negatively charged chloride rushes in, or positively charged potassium rushes out. Either way, the inside of the cell becomes even more negative, dropping from its resting negative 65 down to negative 70 millivolts.
And that drop is called hyperpolarization.
Yes.
It drags the voltage further away from the firing threshold. It actively suppresses the neuron. But the nervous system also utilizes an even earlier intervention called presynaptic inhibition.
This mechanism is brilliant, honestly.
It really is. GABA can actually act on the incoming presynaptic fibril itself before the synapse even connects to the main neuron.
Right.
GABA opens chloride channels on the incoming wire, so the negative chloride cancels out the incoming positive signal before it even reaches the destination. It's literally like cutting the mic cable before the speaker even walks onto the stage.
It is an incredibly elegant control mechanism.
Yeah.
Now, the neuron have to calculate all this incoming data. One single excitatory synapse firing is almost never enough to cause an action potential. It might raise the voltage by like half a millivolt.
And you need a 20 millivolt jump.
Exactly. The neuron achieves this through summation.
Okay, so you have spatial summation and temporal summation. Let me make sure I'm visualizing this math correctly. Spatial summation is when many different synapses located all across the cell membrane fire at the exact same time.
Right.
It's like 50 different people all putting their hands on a car and pushing at the exact same moment to get it rolling.
That's a perfect visualization. The fluid inside the soma is highly conductive, so all those tiny separate voltage changes add together instantly.
And temporal summation is when a single synapse fires over and over again incredibly fast.
Yeah.
Because the voltage change from a single fire lasts a few milliseconds. The next fire adds its voltage on top of the lingering charge, stair-stepping the voltage higher and higher.
Right, building it up.
So that's like one incredibly strong person repeatedly shoving the car really fast before it even has a chance to slow down.
Exactly. And the neuron is constantly summing up both excitatory and inhibitory inputs simultaneously. If a neuron receives a wave of excitatory input, but just misses the threshold, say the voltage only reaches negative 50 millivolts, it doesn't fire.
But it enters a state called facilitation.
Yes, it is primed. It's hovering just below the threshold, meaning the next tiny signal that comes along will easily set it off.
Okay, so this brings us back to the dendrites. We established earlier that the vast majority of synapses, up to 200,000 of them, sit out on the dendritic branches.
Right.
How does the physical shape of this massive tree impact that final mathematical calculation happening down at the axon?
Well, this is where anatomy directly dictates function. Dendrites are long, thin, and their membranes are inherently leaky.
Leaky, okay.
Crucially, they don't fire action potentials themselves because they lack enough voltage-gated sodium channels. Instead, they use electrotonic conduction.
Meaning the electrical current just physically flows down the fluid inside the dendrite, traveling toward the soma.
Yes, but because the membrane is leaky to ions like potassium and chloride, the electrical current bleeds out as it travels.
It loses steam.
The signal physically fades. This is known as decremental conduction.
So distance mathematically matters. A synapse firing way out on the very tip of a dendritic branch is going to lose a lot of its voltage before it reaches the soma.
Right.
But a synapse firing right next to the soma is going to hit the calculation with full force. Proximity gives it a much louder voice in whether the cell fires.
Which brings us to the ultimate question of this chapter. How does this delicate, integrated system hold up under severe physiological stress?
The source material runs through several extreme stress tests. First is fatigue.
Right, fatigue.
If you stimulate an excitatory synapse rapidly and continuously, it eventually just stops firing. But this isn't a failure of the system, it's a protective mechanism. It is a literal exhaustion of the neurotransmitter stores in the presynaptic terminal. When a brain becomes dangerously overexcited, like during an epileptic seizure, it is the synaptic fatigue that eventually forces the seizure to stop.
It acts as an emergency circuit breaker.
Exactly.
Then there is pH. The acidity of your blood drastically alters synaptic transmission. Alkalosis, which is when your blood pH rises above the normal 7.4, greatly increases neuronal excitability.
So let's relate this to a real scenario. Imagine you get so anxious before a massive physiology exam that you start hyperventilating.
Okay.
You are rapidly blowing off carbon dioxide, which causes your blood pH to rise. That alkalosis makes your neurons hyperexcitable. The text points out that hyperventilation alone can be enough to trigger an epileptic seizure in susceptible people.
And conversely, acidosis, a drop in pH, depresses neuronal activity.
Right.
If a patient experiences severe diabetic ketoacidosis, their pH drops significantly. This depresses the neurons so much that they simply stop firing, almost always leading to a comatose state.
Then there's hypoxia, a lack of oxygen. The brain's excitability is entirely non-negotiably dependent on an active oxygen supply.
Oh, absolutely.
If blood flow to the brain stops, the neurons lose their excitability in just 3 to 7 seconds, leading to immediate unconsciousness.
Finally, we look at external drugs. Caffeine directly lowers the threshold for excitation, making it easier for neurons to fire.
Right, we all know that feeling.
But then you have a lethal toxin like strychnine. Strychnine doesn't lower the excitatory threshold. Instead, it chemically binds to and blocks the action of the inhibitory transmitter glycine in the spinal cord.
It cuts the brake lines.
Exactly.
Because excitatory signals are constantly firing a little bit in the background. If you remove the glycine holding them back, those excitatory signals run away completely.
Yeah.
The result is severe, rapidly repetitive muscle spasms that can be fatal.
If we trace the logic backward, we see the entire unbroken chain of this chapter. The microscopic anatomy, like the leaky membranes of the dendrites, supports the function of decremental conduction. That function governs the regulation of the system, how EPSPs and IPSPs summate to reach a threshold. And that precise regulation results in the integrated system behavior we just described, dictating exactly how a human responds to hyperventilation, oxygen loss, or a toxin like strychnine.
It's an incredibly robust, deeply logical system. We started at the macro organization of the billions of neurons in the sensory and motor highways.
Yeah.
We zoomed all the way down into the active zone to watch calcium ions violently trigger the SNARE complex. We watched the sodium-potassium pump furiously bail water out of the leaky boat to maintain negative 65 millivolts.
We covered a lot.
We really did. And we zoomed back out to see how altering that delicate math changes human consciousness entirely.
It truly is a masterpiece of biological engineering.
Before we wrap up, I want to leave you with one final thought to ponder based on this text. We talked about how metabotropic receptors use G proteins to activate gene transcription. Consider what that functionally implies about the last 20 minutes. Every time you deeply process new information like following the logic of this very chapter, those alpha subunits are diving into the nucleus of your neurons, rewriting gene expression, and physically rebuilding the structural architecture of your synapses.
That's amazing to think about.
You are literally anatomically different than you were when this deep dive started.
The physical act of learning is the physical reconstruction of the brain.
We hope this helped lock in the logic for your upcoming exam. Stick strictly to the mechanisms, trust the flow from anatomy to integrated function, and you are going to crush it. On behalf of the Last Minute Lecture team, thank you so much for studying with us.